Um, Thank you. It probably never get used to seeing my name on other people's slides. So what we're going to talk today about is making models, like the previous speaker, of making models of the active form of kinases. I had to change the title because we went from 438 catalytically competent kinases to 437. And what I mean by catalytic kinases are those that can perform phosphorylation, as opposed to pseudokinases, which may be missing one or more of the catalytic residues. And so humans have 494 typical kinase domains, those that look like pKa, the one like the structure that I'm showing here. This is insulin receptor kinase, and 57 pseudokinase domains. So since we're trying to make the active state, we're going to make models of the catalytic kinases, not the non-catalytic kinases. In the protein data bank, what we're looking to make models of are the active forms of kinases that should be able to bind ATP, magnesium ions, and substrate. And that's an important distinction, is that we need to know the conformation of the entire activation loop in order to bind substrate. Having models of active kinases is going to help us understand substrate specificity, the differences between active and inactive conformations of each kinase, understand regulation of catalysis, the druggability of the active form of kinases. Not all inhibitors bind to active forms, but some of them do, and the effect of mutations on kinase activity, for instance, in cancer. And so the question is, can we use AlphaFold to make models of all 437 of these kinases in their active form? But that raises the question is, what's an active kinase structure look like anyway? And how many of them are there in the PDB? And the way we're going to do that is to look at substrate-bound structures of kinases, as well as ATP-bound structures. A few years ago, we published a study on conformational clustering of the first part of the activation loop. And what we did is we took a set of structures that had ATP bound, they had magnesium, the activation loop was phosphorylated. Not every kinase requires having a phosphorylation on the activation loop to be active, but at least if they are phosphorylated, that makes them more likely to be in the active conformation, and decent resolution. And at that time, we had about a dozen structures that we could pick from the PDB, a dozen different kinases that we could pick in the PDB that satisfied those criteria. And those are shown here. The activation loop here is in here in magenta. And you can see how it, here is ATP, but it goes all the way out to the right-hand side and all the way back. And that's a theme that's going to come up over and over again. But if we look at the XDFG motif, X being the residue before the DFG motif, we see that the conformation, the positions of the side chains, the dihedral angles, of all of these atoms are all the same across this set of structures. There's high similarity. So we're going to go have a number of criteria that we're going to use to say that a kinase structure is active. And the first of those is, as the previous speaker noted, DFG-in. And that means that the aspartic acid is near where ATP would be, but also that the phenylalanine is in a certain position, and that position is sort of adjacent to or underneath the C helix. And so on the left-hand side, these are structures of EGFR. The cyan residues are all underneath the C helix. Those are DFG-in. These purple or magenta ones over here are DFG-out. That phenylalanine has popped out of that pocket underneath the C helix. And many kinases have some structures, some positions of that phenylalanine that are sort of intermediate. And so we used a couple of distances of the C-zeta atom of the phenylalanine ring to a lysine of the salt bridge in the N-terminal domain, as well as to an amino acid that's 4 residues after the glutamic acid of the salt bridge that's in the N-terminal domain. So on the right-hand side is a plot, basically a scatter plot of those two parameters. And the cyan points up here are all DFG-in. They are all ones that have a distance where the phenylalanine ring is kind of close to the C helix, but further away from the lysine C alpha. You can't really see them, but the diamonds here are those catalytically primed structures that we looked at in the last slide. DFG-out structures are down here. The phenylalanine is close to the lysine, but far away from the C helix. And type 2 structures are also represented in here as I think circles or diamonds that you probably can't see too well. What we also looked at were the dihedral angles that help place the aspartic acid and the phenylalanine. We started with the dihedral angles that we look at are the phi and psi of the X residue, that is before the DFG, phi and psi of the aspartic acid and the phenylalanine, as well as the side chain dihedral angles of the phenylalanine. So here's the active site here. There are many interactions that are important for activity, for binding ATP and binding magnesium. And there's a certain set of dihedrals that are consistent with forming that active site. And so if you follow along here, if we start with the X residue, the Ramachandran map here, that's these blue points up here. So it's in the B region of the Ramachandran map. Aspartic acid of the DFG motif is in green, and that's in the L region of the Ramachandran map. It's actually fairly high up in the L region. It's not, sort of left-handed, the pseudo-helical sort of position would be here. It's a little bit further above than that. There's a bit of strain in those dihedrals. And the phenylalanine is in the A region of the Ramachandran map. And for extra information, the glycine is also in the A region, but a little bit to the south, southeast of it, I guess, if you want to look at it that way. The chi-angles of the aspartic acid are always trans in these structures, and the chi-1 of the phenylalanine is always G-, is always 300 degrees. And so we did clustering based in dihedral angle space using dbScan and using a measure that's used for comparing dihedral angles to each other. And if we look at all of the DFG-in structures, we can group those into 6 different conformations. The most populated one of those is BLA. So that's the X residue, the ASP residue, and the Phi residue are in the B, the L, and the A regions of the Ramachandran map, as I just showed on the last slide. Those contain these catalytically primed structures. And the magenta color here is to show that the phenylalanine has a G- rotamer conformation. Another common inactive conformation is BLB+. That's this column here. And that's sometimes called SARC inactive by some authors. And that's 9% of the structures in the PDB. And in that case, the naming convention is the Ramachandran names for the first 3 residues, the X, the D, and the F, and then also the rotamer of the phenylalanine. And so that's the plus rotamer for that BLB+. DFG out structures are B, B, A minus. That's the most common sort of set of dihedral angles for those structures. And those are the ones that bind type 2 inhibitors. The next criterion that people have studied for a long time is the existence of a salt bridge in the N-terminal domain. That's between a glutamic acid in the C helix and a lysine in the beta-3 strand. And so this is a selected set of structures that have substrate bound to them, and that salt bridge is always intact. And the lysine chelates part of the negative charges on the ATP. But that's, those are criteria that we've had for a while and that people have used. But what else makes a structure active? And so what we've done is isolated a set of 40 substrate-bound structures from the PDB. And most of those have peptides. And here's sort of a chart of the different protein kinase families and how many unique substrate kinase sets that we have for each of those. We're also including a bunch of protein-protein interactions, including autophosphorylation structures. We had a study on that in 2015, where a phosphorylated an autophosphorylation position of one monomer in the crystal is sitting in the active site of another one, as if it's about to get phosphorylated, and the structure of that site is consistent with phosphorylation. And so we have 40 of those altogether. And what you notice in all of these is that the activation loop starts here in magenta and goes all the way out to the right and then kind of comes back. And that's a consistent structural motif that is required for binding the substrate. And the substrates here are in dark blue. And you can see that in these 4 different families. Tyrosine kinases bind their substrates a little bit differently. They bind them, the other 3 families and all the other families bind them in a groove that's sort of between the 2 parts of the activation loop, the N-terminal end and the C-terminal end. In tyrosine kinases, they make hydrogen bonds actually with the C-terminal end, like a little beta sheet. And so what makes a kinase active? If we look at interactions between these substrate residues and residues at the beginning of the activation loop at the end, what I've done for a selection of these is in red, I have marked residues that have contacts between the substrate and residues in the activation loop. And so DFG, the 4th residue right after DFG, is a contact in almost all of these structures, and then occasionally residues 5 and 6. And so the consensus is that, and there's also interactions with the DFG itself, is that at least these first 6 residues, and not really much more beyond that, have contacts with the substrate. Going from the other end, if the C-terminal end of the kinase has that sort of motif APE, if we look starting at the 4th residue and going as far as about the 15th residue, you see contacts with an awful lot of contacts at sort of 7, 8, 9, 10 back from the end of the activation loop. And so those will help us sort of get criteria for making models of active kinases. And so the next criterion we're going to have is actually a hydrogen bond that we noted in 2019, that there are backbone hydrogen bonds between the sixth residue of the activation loop, that is DFG XXX, that's the third X, and the residue before the HRD motif, which is the HRD motif of the catalytic loop. And so these hydrogen bonds are shown here for the kinase EPHA2. On the right-hand side is structure where that is the structure of EPHA2 where that interaction is broken and the activation loop is kind of folded up and is quite different in conformation and position compared to this one. And so if we look at that in all the substrate-bound structures, those interactions, those hydrogen bonds between DFG6 and the residue before HRD is present in all of them. Now we're going to look at the C-terminal end of the activation loop. These are structures of aurora kinase that are in the BLA minus, so that active, at least at the DFG motif, the active form of the DFG motif. If you look at all of these structures of BLA minus, one thing we notice is that there's sort of two dominant conformations for aurora, one of which looks like it's consistent with binding substrate and the other one doesn't. And so we're going to call those conformation 1 and conformation 2. And you can see conformation 1, the activation loop goes out and it comes back again. The structure is phosphorylated. In conformation 2, it goes out, but it comes back and it's kind of got this sort of double hump thing. And those structures are also phosphorylated. If you superimpose these aurora structures on structures that have substrates, you can see that the substrates fit, and they're in light pink here, with a bunch of other CAM-K kinases and AGC kinases. The structures of aurora in conformation 1 are consistent with substrate binding, but the structures in conformation 2 are not. There's a very large clash here between this sort of double hump and where the substrate would be. And so these are substrate capable and these structures are not. And the reason those are substrate capable is because of the structures where this conformation 1 have TPX, a protein called TPX2 bound. And what TPX2 is a disordered protein, but it binds to aurora and activates it. It's known to activate it experimentally. And what it does is it sort of yanks on the activation loop and kind of pulls it outwards into the structure that you see here on the left-hand side. Whereas in the absence of TPX2, it's like a spring or a slinky. It's all kind of bunched up in a conformation that's not consistent with substrate binding. And so the distance that we came up with to try to identify which ones were active and which ones were inactive is the 9th residue back from the end of the activation loop has a fairly short contact in most of these substrate-bound structures with the carbonyl oxide, so it's the C alpha of APE9, this 9th residue back, and the carbonyl oxygen of the residue of the arginine residue of the HRD motif. So here it is in an aurora structure. So if we count back PPE all the way back to this glycine, that's that interaction that's 3 angstroms right there. Here it is in pKa, also a short distance, also a glycine. But here it is in that conformation 2 of aurora, where the distance is something like 10 angstroms. And you can see how sort of pulled away that the activation loop is from that short interaction, and where it would clash with the substrate, where the substrate usually binds. So here it is in all those substrate-bound structures. Here's this short contact of 3 or 4 angstroms between the carbonyl oxygen and the C alpha of that residue. So if we're going to answer the question, how many active structures are there in the PDB, we're going to use these 5 criteria: the DFG in, BLA minus, the salt bridge in, a distance of 3.6 angstroms, the activation loop N-terminal criterion, that's the 6th residue of the activation loop, with a hydrogen bond distance of 3.6 or less, and then the C-terminal contact that I just described. So that's residue 9 to the HRD, and we're going to use a cutoff of 6 angstroms for that. So if there were 437 catalytic kinases, only 268 are in the PDB at all. 202 of those 437 are BLA minus. But if we start applying the different criteria, especially that C-terminal criterion on APE9 residue, only 162 of those 437 structures are actually in the PDB in an active form, or at least that satisfy that requirement. And so if we require all of these things together, only 155 are actually available in the PDB out of 437, that's 36%. And if you want to look at the whole activation loop being present, that is having coordinates, it's often disordered, that number is down to 30%. And so we're going to do what other groups have done, which is to use specific templates that are in the active form as a way to make active, as a way to force AlphaFold to make active models, and then shallow sequence alignment. So Ho and Feig did this for GPCRs, as well as Del Alamo did it for GPCRs and kinases. And so the sources of the multiple sequence alignment are going to be sets of orthologs. We got all of the kinase sequences from UniProt. For every kinase, we picked the top 25,000 closest sequences, and then only chose as orthologs or maybe close paralogs kinase sequences that were more than 50% identical to the query. So they're relatively similar to the query. And they covered 90% of the kinase domain. And then we use CD hit to make sure that none of the sequences in our ortholog sets for, let's say, PKA, for one specific kinase, we made sure that they were less than 90% identical to each other. If you just use the top 50 hits for EGFR through UniProt, they're all going to be 98, 99% identical to EGFR because every mammalian sequence of EGFR is very similar. And so you need to sort of pare them down. We also use sequences in the same kinase family. That is, for modeling an AGC kinase, we're going to use just all the AGC human kinases of 60 or so kinases. And then we also ran UniProt 90 just as a comparison. And then the number of sequences in the MSA, we tried everything from 5, 10, 15, 20, up to 90, and then 10,000 means just using AlphaFold in its default method. For the template databases, we used these 5 criteria to identify active structures in the PDB, and we made a set of 165 kinases and 278 chains, both human and non-human kinases, we allowed those to. And then we used something that we call distillation templates. Those are active structures, structures that we built with AlphaFold with this procedure that produced active models. And then we just use those as templates for making models of other kinases that we don't yet have an active model of. So we call those distillation templates in reference to the original AlphaFold paper, had a distillation set of models that they used for training. And so this is what happened. So let's just look on the right-hand side. These are for the ortholog sequences. The x-axis is the MSA depth, running from 90 sequences all the way down to, we actually tried 1 and 2 just in case. And so as you can see that the lower, the less depth there is in the MSA, you get more active kinases. So the y-axis is the number of kinases out of 437 that we managed to make active models for. And so, by the time you get to sort of 5 or 10 sequences, you're getting active models for 410, 420 of the 437 kinases. No one method, no one choice of template set and MSA set made models, active models for all 437. However, if we combine all of those different sequence depths and different template sets together, we got active models for all 437 kinases. There was one kinase, obscurin-2, and you could tell by the name obscurin that it's going to be a tough protein. We had to make a mutation actually in the activation loop to get an active model. It's possible it's a pseudokinase, we're not really sure. But this is what the models look like. So for 51 AGC kinases, again, we see this motif of the activation loop starting here, going all the way out to the right, and all coming back. And they all have this kind of upside down, round bottom boat at the C terminal end of it. That residue 9 is about here where my cursor is. Tyrosine kinases are different. There's actually much more variety in that C terminal segment. And so to pick the best model out of all the models that we make for every kinase, what we do is we measure the minimum value of the PLDDT of all the residues in the activation loop. So just across the 30 residues in the activation loop, what's the minimum PLDDT, and then we assign that as a score of the activation loop. And so this plot is the minimum PLDDT on the x-axis. On the y-axis is the RMSD to a substrate-bound structure that we use. So for AKT1, you can tell that by the time you get to the highest PLDDT, you're down to a RMSD of about 0.5 angstroms. And the structure superposition of those, of the model and the active structure are here on the right. Same thing for CDK2. Again, some very poor PLDDTs had rather bad RMSDs, but the best PLDDT had a very low structure. If we benchmarked it against 130 structures from the PDB that had full activation loops that were active, and 80% of them have an RMSD of better than 1 angstrom, 90% have a better than 2 angstrom. And so this is the distribution of the RMSDs. We found some outliers that we made poor models of, and it turned out some of those we think are not, that pass our criteria as active are not actually active. So here's a structure of Sark human. There's a funny little turn here. The Sark chicken structure, however, looks just like our AlphaFold models. And also those structures look just like ABL1, which is, here's a substrate-bound structure. So some of the structures that that pass our criteria that we think are active, if you look at the details of the activation loop, some of them are not. So just to summarize, we use these 5 criteria to identify active structures in the PDB and then also applied this to the AlphaFold models. We can model all 437 in an active form. Inactive states are going to be more complicated, which we're working on that. And then we can use these models to bind substrates and to model other sorts of interactions. And then finally, somebody turned 60 years old today, as of about a half an hour ago. I look more like my baby picture than most people do. I still have the same haircut and the same weird expression on my face. This was all work by Bulat Fayazov. Vivek did all the earlier King-Core work, and then here's the website. I am active on Twitter, now called X, and I tweet about structural bioinformatics. I rant about people who park cars in bike lanes. I rant about bad COVID takes by Nobel Prize winners. You all know who I'm talking about, or some of you do. And I tweet about LGBTQ issues in science. And my partner Sam is in the audience. He's taking me out to a fancy dinner tonight for my birthday. So thank you.
Thank you, Roland. Happy birthday.
Thank you.
And thanks for being a role model for me and many others. Do we have some questions for the audience? There. Sorry, some new face for questions. You made me run. You could help me a bit.
Great talk, Roland. Really cool how you establish the criteria to ascertain which structures are active, and then for result what took AlphaFold to generate those confirmations. My question is, you said you selected the best models with the minimum PLDT in the loops, and I was wondering if you, because sometimes I try to do this, and I wondered if you tried average, and if you decided to use minimum, and if you examined what were the differences.
We did try average, and I can't say we did a really head-to-head comparison to see like which one behaved better, like on this benchmark. This benchmark is kind of recent, this 130, and it would be interesting to try the average one. But we really wanted to make sure that the entire activation loop was modeled. So we didn't. If you use the average and it's a long activation loop, you might have some residues in the outer part that have very poor PLDT. But you're right, it's certainly an alternative. And also just as listening to people in the last couple of days is we could use the PAE values too of the activation loop residues with the rest of the structure and to see if that would be helpful in finding what's the best. The thing is that there's still conformational diversity in the PDB with active structures and with things that we think are active that look pretty good, look like a bind substrate. There is still some conformational diversity in that outer region, the furthest away from the ends. And that's probably real, and those structures are probably all capable of binding substrates. So we're not going to ever get everything to be 0.5 angstroms for every single kinase.
Great, and a quick follow-up. Are the minimum PLDT always in the middle of the loop, or? Usually, yes.
And with this, we have the last question from this 2023 edition of 3D-SIG there. I will lose weight.
Thank you. Thank you for your talk. Very nice. And congratulations and happy birthday. I have a question regarding pseudokinases. Can you use this approach, try to identify these pseudokinases that may be active?
Sorry, what kind of kinases?
The pseudokinases. Pseudokinases.
Oh, pseudokinases. Okay. We can use the same method on pseudokinases, and we can use the same criteria. And a lot of pseudokinases do have conformations that look active. They look like they could bind substrates. They don't, we don't think. And some pseudokinases have what we would call inactive forms and active forms. They are a conformational switch. And we decided while we're looking for active models, we should just focus on the real kinases, on the catalytic kinases. But the pseudokinases present another interesting problem in seeing what kind of structural diversity they have in the PDB and what kinds of structural diversity that we could calculate with AlphaFold. That's a good question.
Thank you very